Okay, so thank you for the opportunity to present the research of part of the research of our group and making a conference in such a great place. The title of my talk is quite general, role of 3D genome structure in the regulation of gene expression. It is a topic of interest of several groups all around the world. And I will focus on one specific result we achieved last year. And since not all the people in the audience are working on genome structure nor even on genetic gene expression, I will start with a few general facts. First, this is an old picture. It is a picture from a review paper by Helmut Schissel, and it is an artist's view of our understanding of the genome architecture about 15 years ago. So it is a good slide to present to you the different objects from DNA to chromosome passing from the chromatin fiber, which will be the main actor of, in my talk. And a few terminologies: the nucleosome, which is the wrapping of DNA around histone core, 8 proteins, and this nucleosome is a very conserved entity which plays an important role in all the genomic architecture. So 15 years ago, this was a simple picture. You have DNA wrapping around histone core to form a bead and string fiber of nucleosomes. This bead and string fiber itself wraps onto itself, forming the chromatin fiber up to the chromosome in some debated and not very investigated way at that time. So the main point at that time was to understand the interplay between DNA and chromatin, how DNA and nucleosome architecture were forming the chromatin fiber, and conversely how the presence of a chromatin fiber modifies the function of the DNA. 15 years later, so that is very recently, about only a few years ago, the picture has changed quite significantly by the addition of a totally new level of investigation, the supranucleosomal scale, in between the chromatin fiber, here, the nucleosome and the chromatin fiber, and the chromosomes. So this new scale of investigation appears to play a real important role in the regulation of gene expression, and it is the focus of a lot of investigation, both on the theoretical side and an experimental side. And what are the main experiments that give access to this level? It is a whole set of experimental techniques termed chromosome conformation capture. So the idea is quite simple. The realization is more difficult, but you could understand quite easily how it works. The first step is to extract living cell from a tissue, for instance, mouse, a human, to isolate the nuclei, just to avoid mitotic state, so intact nuclei, and then to cross-link the genome, the contact across the genome. So you see, you have, I should not escape from the field, so I will take that. The genome is something like that. Inside the nucleus, it has some very complex conformation, and this chemical protocol is able to cross-link any contact you have in the genome, in living cells. So this is what is represented here. So any loop, any closeness between two genomic sites is fixed at some point. Then additional steps of digestion, ligation, inverse cross-linking, and then sequencing allow to identify by matching to the genome sequence which genomic sites are in contact in a living cell, in the nucleus of a living cell. So there are several variants of this protocol, from local, very quantitative ones to genome-wide one. And the result is the identification at a given moment in a population of cell nuclei of the pairs of genome sites which are close one to the other. The data we are investigating in the group with Julien Mosiconacci in Paris is what is called IC data. It is genome-wide data. The counterpart of this genome-wide range is the moderate resolution. At that time it was 50 kilobase. Kilobase, it's a 1,000 base pair. And now, in some condition, it is possible to reach 1 kilobase, but with some balance between resolution and statistics, and I won't enter into this detail right now. The result of such experiments is what is called a contact map, like a matrix, or contact matrix. Actually, it is a matrix, where you have on each axis the genome location, linear location along the genome. So here it is successive. You can imagine that you have here the stretch of DNA with a different position along DNA. Here the same. And like in any matrix, something here is the contact frequency between this site and this site. The diagonal is, of course, the trivial contact due to closeness. You have, and for instance, you see various structures, small-scale structures, larger-scale structures, and so on. So this is a matrix representing the frequency of contact, the probability of contacts between any pair of genomic sites at the genome-wide scale. Here, the picture represents only the chromosome 1, but you can have all the genome in such maps. So this is a huge set of information, presumed to be very useful. You can do some motif analysis right on these matrices, on these maps, and extract already a lot of information just by the statistics, which are the region which are the most connected to the other one. Do you see some modules? Do you see, so there is a lot of analysis directly on these maps. But perhaps it is your impression, if it is the first time you are seeing such a map, it is not really a visual view of the 3D shape of the genome. So we would like, to complement the statistical analysis of motifs in such maps, to really see what is the underlying 3D shape of the genome. So this is a huge challenge, and we proposed an algorithm to do this reconstruction. That is, to extract the coordinates, the 3D coordinates, the shape in space of the genome from the knowledge of these contact maps. So just here to be specific, you have 4 matrices involved in this problem. What we want, the coordinates, what we have, the contacts, and 2 intermediary matrices, a distance matrix, that is the distance between any 2 pair of genomic sites, and another auxiliary matrix, which is termed the Gram matrix, and which is simply the matrix of the vector products between the different coordinates. That is, the component ij is the vector product between the coordinate vector of the point on the site I and the coordinate vector of the site j. So we have those 4 matrices, and it is quite straightforward to pass from the coordinate vector to the Gram matrix. It is simply a vector product. To the coordinate to the distance, it's simply the expression that we all know about of Euclidean distances. From distances to contacts, You just simply choose a threshold, and all the distances enough short will correspond to a contact. The challenge is to do the reverse way, that is, from the contact to the coordinate. And we propose the red path, that is, passing from contact to distances and then to distances from coordinates. And I will briefly detail those two, this path, the red path. The first step, from distances to coordinates, has been solved in mathematics quite a long time ago, with only a few details to adapt to the present situation. And it is the articulation of two domains. One is, which is a really abstract, pure mathematics, distance geometry, where there is at least a few theorems, very powerful, giving the condition to pass from a distance matrix, that is a matrix where each component is a distance between 2 points, to the matrix of the coordinate. The step is a bit complicated. You pass from the distance matrix to an intermediary matrix, which is a metric matrix with this complicated expression. And under this mathematical condition, meaning that the matrix has a specific property of being semi-definite positive, then you can ensure that it is possible to reconstruct the Gram matrix, which happens to be simply equal to this metric matrix. So this is the first theorem, which is a real exact result. And an approximate whole rank of approaches term multidimensional scaling, which allow to truncate the metric matrix in case where it is not semi-definite positive and get the best approximating structure in a given dimension. So you have the exact mathematical result allowing to pass from a distance matrix to the Gram matrix and then to the coordinate vector, and an additional approximation step giving the best approximation of a given dimension, which is here 3. So with those two part of with those two, I don't know how to say, those two approaches coordinated, linked together, it is possible to reconstruct the structure up to a rotation, symmetry, and dilation. So you pass from the distance matrix to the coordinate just by diagonalizing this matrix. So it is very simple, very straightforward, very efficient. And this is very interesting because the distance matrix in case of a genome is really huge. It is tens or even hundreds of thousand sites. So a matrix of square thousand sites. The most difficult step was to pass from the contact map to the distance. I've explained how, and it's quite obvious to pass from the distance to a contact just by thresholding the distance. The reverse is more complicated because what is obtained in the experiment is only contact, that is, sites of the genome which are in close contact. You have no idea of the distance between sites which are a bit further away. The idea is to consider the contact map as an adjacency matrix of a network. So you have here different nodes. The color is just for explanation. You have the sites. It is simply genome sites with no a priori differences between them. And a link corresponds to a contact. So it is a sparse network because the experiment does not give significant contact between any pair of sites. Some sites are not in close contact. And So you have this sparse network. And the idea is to consider, for instance, for the distance between these white and red sites, the number of steps connecting them. So here, the distance between the white and red nodes will be 2. The distance between white and this red one will be also 2. Between the green will be 3, and so on. So just by taking the topological distance on this graph, on the graph associated, directly associated to the contact matrix, you can reconstruct a distance, a real distance, because triangular inequality is satisfied. We improved a little this basic idea by considering a weighted graph, not weighted in the usual sense, but rather by endowing each link with a length. And the length of each link will be taken equal to 1 over the frequency. The more frequent is the contact, the shorter is the distance. So it is quite a shortcut, but it has been validated by experiment, for instance, direct observation of the distance between sites, which can be done on a local scale. So this is a good approximation, good ansatz, which is taken in different contexts, which has been validated in several contexts. And we have now the same idea, the shorted path, but now the links have not the same length. So for instance, the distance between this white and red site will be the distance, the sum of the links. We tested different ways, so either the sum or the square, the analog of Euclidean distance, so summing the square, the root mean square sum of the links, it does not change a lot. In fact, it does not change at all the result. So we have in this way a manner to reconstruct the distance between any pair of sites, even those which are never in contact in a cell nucleus. And we thus turn the contact map, the sparse contact map, into a complete distance matrix. To validate this algorithm, which is very fast because it involves only diagonalization of a matrix and this the shortest path computation on a network, no optimization, no simulation, no learning and several run of learning steps. This fast algorithm has been tested on simulated data. So this is a simulated yeast nucleus, and then we derived first the direct step, passing from the structure to the coordinates to the gram matrix, to the distance, to the contact. So here it is a direct path, like that. And now it is a reverse path, and we got a very good correlation coefficient, a very good reconstruction. Of course, we tested that on numerous different structures and different conditions, but it appears that for such compact structures, the reconstruction algorithm is very good. This is, for instance, the picture of the human chromosome, all the human chromosome. Each color is a different chromosome. And this reconstruction can be done also at different scales. So this is the full genome, human genome, with all the chromosomes. Each square here is a chromosome. You see that the contact inside a chromosome are far more frequent than the contact in between different chromosomes. And you can zoom. So here it is the zoom on the first chromosome, and you can again zoom. So this is only a part of the first chromosome. This visualization matches other results that have been obtained by direct analysis, but give a visual view of, for instance, the active and inactive compartment in the genome, or of topological domain, which is a modular structure of the contacts. So here you have the genome. It is the diagonal. It is a view where you have this matrix. You just see it on the diagonal, just rotate by 45 degrees. And you see those modules which correspond to the different blobs you are seeing here. This is back to this view. We have now an ongoing project on the nuclear bodies, those entities which are protein complex with some DNA associated. It is possible, we have a technique in our lab, in the Montpellier lab, to extract the sequence associated to nuclear body, and the project is to see which, in which loci, in which region of the 3D architecture those sequences are located. So it is possible to put any result, molecular genetic results, molecular genetic experiments in a 3D context. For instance, another ongoing project is to visualize the epigenetic patterns in the 3D context. Just a brief explanation. Epigenetics is all the information which is on top of the genetic sequence. Here I mentioned the histone modification. So the histone is a protein on which DNA is wrapped into a nucleosome. And there is a lot of possible covalent modification of these proteins. And they have been demonstrated to play a huge and very important role in regulation of transcription. And a lot of big projects, the ENCODE project and several other ones, provided annotation, linear annotation of these epigenetic marks in the form of linear epigenetic profiles. And it is now, with our algorithm, possible to visualize these epigenetic profiles in their 3D context. For instance, this is an overlay of considering two types of histone modification, acetylation, which is presumed to be associated with active chromatin in green. So you see how it is located in the 3D structure, and in pink, the 3-methylation of histone, which is associated to silent chromatin, to silencing of gene. And you see it, the complementary location in the 3D architecture of the genome. So it is possible now with this tool, with this algorithm, to visualize any linear annotation of the genome in the 3D context. This is in human. Human chromosome. I think this is the data from, I think it's stem cell.
It should vary between cells, right?
It should vary between cells, exactly. So this is, I should have the reference, I should give you the reference because we tested that with a different, so it is a data by Calor, and I think it is a lymphoblast, if I do not mistake, because there are Dixon data on stem cell, and so this is just an ongoing project. It's not yet a result. It was just to present you what is our ongoing investigation. Of course, this epigenetic profile vary according to cell types. They vary according to the cell state, because there is now data on epigenome, which has been obtained in different pathological conditions. And the aim is, of course, to correlate changes in this location to either changes in occurring during cell differentiation or changes occurring during, for instance, cancer progression.
Can we ask questions during the talk?
Yeah. Please. I think it basically I was finished.
How precise is it? I mean, could we see slight changes that way? Or?
It depends on. Theoretically, we can accommodate any any data, any resolution. It could be at the base pair scale. It could be in single cell. It could be in population of cells. We can reconstruct, visualize a 3D structure underlying the contact map given to us. So the limitation is experimental. The standard data, because it's very long and expensive experiments, so people are still using quite old data, which means 4 years or something, or even 3 years. And the first one, the first data was have a resolution about 50 kb for the genome-wide set, which is quite poor for such visualization. The recent data now reach 1 kb resolution, which is better. We expect, of course. Actually, it depends on two points. The first one is the resolution of the chemical experiment, of the cross-links, the ligation step, the digestion step, which can, in principle, give a quite high resolution. But then you cannot sequence any all the DNA fragment of your sample, so you have to choose some threshold, considering that anything below is noise. And then, according to the sequencing depth, you get a more or less large resolution. So even if you have experimentally a resolution below 1 kb, if you want a genome-wide map, you have to bin your experimental fragments into a coarser view to get a good statistics. So there are alternative methods, quantitative methods, which are more local, but then you lose the genome-wide picture.
I mean that if we have situation A and situation B, you have a way to compare the two maps on a genome-wide basis and identify regions which are really mobilized by the...
Then you can go at a finer scale with a more quantitative experiment. I can show you. So this is, for instance, a 3C qPCR, which is developed in the group where I'm working in Montpellier. So now it is not genome-wide. You choose a reference point and you investigate all the contact frequency with the different site downwards, and you get the probability of contact between the reference site and all the following one. This is not genome-wide. It's quite a restricted range because it is only up to 500 kb, but now it is quantitative. And very local, you can, basically, there is almost no limitation. So you need, to such a huge question, you need complementary approaches. You need modeling, you need various experiments, you need simulation. So it is really this view, of course, is not the solution to how epigenetic marks will, modification in epigenetic marks will induce cancer. It's just an additional new view giving the 3D perspective on this mark instead of only a linear, broader view. So, but then we have to do, for instance, colocalization analysis, but of course colocalization does not mean interaction, even the contact. This is perhaps an important point which is quite underrated. No, it is in this way. You see here, this would be the contact only due to random collision, due to the thermal motion of the chromatin fiber inside the nucleus. If you do such experiments, 3C experiment, you see some peaks corresponding to what is termed specific interaction. Typically, they correspond to a random contact which is stabilized by interaction between proteins. So this is those two parts in such 3D architecture. There is the physical constraints controlling the big, large-scale moves of the genome, allowing some site to be sometimes in contact, more or less in contact, and then when the sites come into a close contact, you can have the involvement of specific proteins and then a specific interaction stabilizing the random collision. So this is this interplay between the generic physical property of the chromatin fiber and the flexibility, for instance, constraining the shape, constraining the possibility of some sites getting close one to the other. And you have the involvement of specific partners, specific proteins, bound proteins, which create what can be called interactions. So there is a difference between random contacts and interaction, and in such experiments you get both. And then you need some more detailed and quantitative experiments at a more local scale to separate what is due to the physical feature of the chromatin fiber or even larger organization, and what is due to the involvement of specific complexes at a given moment. And so this is a very interesting interdisciplinary subject, because you need both very accurate and fantastic experiment, because it is done in living cells. So it is actually the conformation of the genome in a living cells, not in a test tube nor in your mind. It is nor in simulation. And you need also some mathematical, physical, bioinformatic tools to just make sense of such experiment, and also disentangle what is a real information, what is an experimental biases, and so on. So I will finish by thanking the two teams where I'm working in Paris Laboratory of Theoretical Physics with all the members, and especially Julien Mosiconecci, with whom I developed this algorithm, and in Montpellier, a group of molecular genetics doing the experimental part of our work, and Thierry Fournet, which is the leader, and I'm part-time in both teams and trying to match theoretical physics with molecular genetics on a daily basis.